we'll get ahead and try and introduce our panel. And I've got some discussion points and some of the questions I've kept up as general themes to kind of attack with that. So first, can I get my panel to maybe activate their cameras? And I'm going to do, say you've met many of the panel so far. Dr. Feldgarden at NCBI, Dr. Tsang at LSHTM, Inês Mendes at University of Lisbon and Molecular Medicine, Dr. Andrew Page at the Quadram Institute, Dr. Emma Griffiths at Simon Fraser University, Dr. Carolyn Johnson at JPIAMR, Professor Mark Pallen. Are you what? I don't actually know your current affiliation, Mark. You can jump in on that if you wish. I'm
at the University of East Anglia and the Quadram Institute.
Good. Didn't want to cast aspersions by giving you the wrong affiliation. Professor Henk Den Bakker at the University of Georgia, and Dr. Anthony Underwood at the Center for Genomic Pathogen Surveillance. Welcome. Thank you all for attending. A few discussion points, and I think we'll take the approach of I will throw out the questions and feel free to jump in or put up your hand to take or tackle one. And I will be picking on some of you, especially with when I ask the question. So I think one of the themes that came up today, and we did provide some answers to it. Okay, I have AMR genomics. Which database and tool do I use? How do I know which one to use? I'm starting with the easy questions. Okay, Anthony, you jump in.
I think it's almost an impossible question to answer. And in the project I was involved with, I thought long and hard about which database to use. And at the time, this was about 3 years ago, I actually ended up having a bit of a hybrid solution where I took the acquired database from NCBI. And at the time, the point mutations weren't as well represented in the NCBI database. So I took the point finder database from the ResFinder crew and basically wrapped them up and used Ariba software to merge those things together. And I still, to be honest, find myself in a very similar situation today, is that each of the databases has their own benefits. Each of them, some have better metadata. ResFinder, for example, tries to have a sort of in silico antibiogram. NCBI has really nice release schedules, which I really appreciate, and really well described releases. And so I still find that actually often the best thing to do is to run several databases with your favorite tool or with a tool which uses that database, and to compare some of those outputs. I still don't find there's one database to rule them all, unfortunately.
So I think a question, unless anyone else wants to jump in on that one, apart from harmonization is one way of comparing, and charmed, as Inês talked about. Why are all the databases just little beta-lactamase genes?
Oh, I'll jump in if that's okay.
Yeah.
So one reason is that they were, if you go back to the 80s, there is actually the same kind of crisis we have now, which is the existing beta-lactamases were all failing. So there's been a lot of work put into that. The other thing is, if you're familiar with beta-lactamase nomenclature, you'll see things that are like, TEM1, TEM2, right, and so on. That was actually intentionally designed. George Jacobi told me before he retired, that they gave those names rather than just calling it, another class A or another class C beta-lactamase, because they actually wanted people to have an incentive by giving it a somewhat sexy, at least for biologists, name to get it published. Because the goal was, if you did that, then people would have an incentive to do the biochemical characterization. So there's actually a large community, I think, both in terms of the drug development side, they're effective antibiotics, they're well tolerated, as well as just there was some sort of conscious, subtle efforts to build a community of researchers who know each other. And so that's sort of taken off. That's my take on it, those two. And also because these are curated by, they used to be curated by Leahy, now it's curated by NCBI at their request. Those are sort of, once you have that, people will submit And so you get that information. I guess it's if you build it, they will come kind of thing. So that's my take on it. But
yeah, great. Yeah, and in some ways, I think the other aspect is it's one of the more obvious types of AMR resistance. It has a clear mechanism. It destroys a beta lactam. It's easy to follow in that respect. Whereas when you're getting to more nuanced bits of target protection or target modification, like becomes less obvious. So we're better at studying it as well, I would say. And I'm gonna target Kara for this one, as they have a lot of expertise in this particular problem. How far can we push AMR prediction of phenotype from genomes? There's lots of statistical models, there's supervised machine learning. How far do you think we can push them? And do you think there's limits to what we can actually do with that?
Yeah, so this is another challenging question that I don't necessarily have the answer to, but we have done sort of rules-based methods, statistical modeling, and machine learning methods to try and predict AMR phenotypes. And we have found that for some things like E. coli, statistical modeling like logistic regression worked quite well for predicting phenotypes. But then for other species like Pseudomonas aeruginosa, it becomes much more difficult and very, very complex. And so I don't really have an answer for how to progress our prediction ability for Pseudomonas aeruginosa. I'm not sure if there is a machine learning algorithm that can really learn all those complex mechanisms, but hopefully time will tell, and with more work and the mechanism elucidation being done, we can sort of push towards that. But so there is no right answer. And then for some organisms like Klebsiella that we're working on now, we're thinking that maybe a rules-based method could work. And so that's like on the simpler end of things. So, yeah.
Do you think there's the getting a good prediction of phenotype isn't necessarily always the end goal? Is there sometimes, do you think there's utility in just making some of these models try and learn more about the mechanisms?
Yeah, so what we've shown in our work is that sometimes we can find new phenotypes for already known resistance genes. So we look into the models and then we find which are the strongest predictors, and then we phenotypically test them using experimental validation. And then we found that these known resistance genes actually have phenotypes that we wouldn't necessarily expect or haven't been published before. And so I think that it is still a useful exercise to see which mechanisms are really driving the underlying resistance of these bugs. Great.
Anyone else want to jump in on that? Or I think it was pretty comprehensively covered. Okay, what about AMR genomics of difficult aspects? Do we tend to just ignore repeats and mobile genetic elements when we can't assemble them very well? Are there any bioinformatics tools that are better suited to dealing with them in relation to AMR? Andrew, you're nodding and shaking your head. That means you're volunteering.
Yeah, I think fundamentally, if you are using short reads to look at highly repetitive AMR genes, where they've come in repeatedly over and over again in the same place, that's a technological limitation, and you just need to use a different technology. You have to use longer reads. But that is one of the great things about, say, Nanopore PacBio, that we are being able to explore these things. We're not just seeing, massive copy numbers and collapsed repeats and missing all the information that is in there. Is it 10 copies? Is it 5 copies? Who's to know? So long read metagenomics really will help. Long read sequencing of isolates will help, and long read metagenomics will help even more. So I think you have to use the right tool for the right job, and there's no point in just messing around trying to, make a round peg fit in a square hole kind of thing, or probably got that the wrong way around. But so bioinformatics can only do so much.
So on that topic, what are the tools for doing AMR analysis from long reads?
We need to invent them, more or less, I think a lot of people just take tools for short reads or for assemblies and then try and apply them. But of course, at the moment, you've got errors, particularly in just raw PacBio data, which need to be taken into account because, if you have a single SNP or a partial gene, is that really there or not? You know, is that a functional or not? You know, unless you have the actual phenotypic data to back it up, you don't really know. And then, of course, there's all the other resistances where we can't explain it, or intermediate. Like I absolutely hate when someone says intermediate, you know, but they don't tell you, you know, what the MIC is or whatever, and then you're lost at that point.
That's always inevitably the tension in bioinformatics and life sciences in general is newer technologies give us the prospect of getting better answers, but we haven't written the tools to use that new data very well yet. I
think we just need an army of bioinformaticians employed for the next 10 years to write all these tools.
And maybe some of the attendees here can be a part of that group.
Absolutely.
But okay, there are a lot of different tools and approaches. And one of the biggest things they vary in is sensitivity. So can they detect anything that even looks like an AMR gene? Do they capture all of the AMR genes that we know about in that sample? Or specificity? Do they capture, you know, are the ones they capture actually AMR genes? So on one end, we have highly specific things that people talk about, like PCR, where we're hopefully, if the primers are designed well, very specific in identifying, okay, we found this one particular beta-lactamase that we designed primers for, we got that out. On the other hand, we have potentially sensitive methods like metagenomics that can look at potentially all the AMR genes in an entire ecosystem with sufficient depth and lots of caveats. Like, how do you choose based on your particular domain where to land on that sensitivity and specificity spectrum? Hey, come on. I know some of you work with, especially with public health and some of the questions about this.
Yeah.
Just
go ahead, please.
Just a quick, I mean, our approach, because we actually work with other U.S. agencies that are involved in public health and surveillance. So we are typically very cautious. We try to err on the side of being conservative in terms of, or at least if we're going to say, well, this gene was only hit with, let's say, an HMM, that we make it clear to the user that that's the case. So we err on that side because we're not really looking at gene discovery. You know, we're not trying to do, you know, here's this novel gene from this organism. We're really trying to help them make some very specific decisions about even regulatory issues, or, you know, is this a problem, that kind of thing. So we're somewhat conservative on that in that sense.
I guess another huge problem is that we're highly biased in what we do. You know, we just look at, you know, these human pathogens which cause disease, and I know those are and sometimes animals, but I know these are the ones that people target with antimicrobials. But there is antimicrobials out there everywhere, you know, in the soil, if you look at bacteria there, whatever, and we're missing so much of the actual knowledge because we're only targeting, you know, pathogens which make up a teeny tiny bit of what's actually out there in the environment in the real world. So I think we do need to go on a big fishing expedition and stamp collecting and look at more than just pathogens which cause serious disease, you know, because at one point wasn't half of NCBI just E. coli K12, might be going back a bit, but you know, unfortunately, when you have things that are so biased, it can overwhelm databases. You know, when you're like, say, Salmonella, you know, there's so much work, say, done on Salmonella typhi, which is MDR and whatever. But then, you know, what happens when you look at a different Salmonella? You know, are we missing things in Salmonella that doesn't cause as severe disease? Carolyn.
Yeah, I actually I think that's a really fantastic approach. But as a funder and as someone completely outside, we're also really interested in looking outside of the bacterial world. You know, there is a whole ton of different resistance mechanisms out there, and we're only just beginning to scratch the surface with understanding bacteria. But yeah, I was talking about this just yesterday, and they were saying, well, you know, can we adapt these tools to look at viral resistance? You know, is there a way we can do this? And I think it's totally laudable to start looking outside of just those pathogens that cause human disease, but really also would be amazing to extend that fishing expedition further and start looking for different things in different pathogens. So that's a scary thought, but that's from the funder point of view.
And the funding is actually one of the major difficulties with a lot of this work. Is it's very hard to get the funding and resources to even maintain these big databases. Even, I'm sure, even Mike has had to fight internally with an NCBI and NIH at times to keep like resources funding, and that's in a relatively well-funded organization compared to individual labs and groups that are running some like the ResFinder and CARD database. So there's a lot of ambition to extend these databases, but there's also caution, because if you try and add all of antifungal resistance to your database, all of a sudden you've got to support that as well. And there are real-world implications for not curating that data very well. And it potentially is going to be used somewhere, and there can be human cost, which is something in bioinformatics that we get in the pathogen side, but other parts we just never even think about the problem, like bugs in our code actually having a real-world impact on someone's health. But yeah, so it's maintaining these databases. So they're always keen for people to volunteer and contribute if anyone does have an interest in one area. And I know most of the databases will have expert curators in certain areas, certain gene families, certain species that do help them contribute and contribute to that curation of different mechanisms, etc. Anthony?
Yeah, you're making a really good point actually about the funding of databases, because I think we've always found this within the public health realm as a whole, is that funding comes more easily to the kind of like the interesting research topics, but databases never really garner that much interest. But actually, they're the most crucial things that we can have. Really, is reliable, well curated, frequently updated databases. And so, I think it's still a dilemma: is how do we find funding to fund those crucial databases on which not only research depends, but increasingly clinical action and surveillance will depend upon. So a big call out to funders, the big funders out there, please fund databases as well as cool research.
We really need to, and I think it's something we're totally aware. It's absolutely essential that we really start to look into properly funding these kind of underpinning resources, because without them, you guys can't, the interesting research can't happen. And more criticisms of grants that are interesting research is your data's not, you haven't got a good plan for curating your data, or you haven't got a good downstream data analysis plan. So really essential. And yes, I totally agree. And we are thinking about it, looking at it, trying to figure out a solution.
Can I just tack on to that? So it's really hard to get sustainable funding for databases. It's even harder to get money for standards and ontologies and for curation that are absolutely critical to making sure what's in those databases are high quality. So yeah, that part I think needs to be a little bit higher on people's radar as well.
Absolutely, it's essential.
And it's a challenge, because while databases and curation have massive impact, they're some of the hardest impacts to measure as well. So even if the funders are beholden, the people actually doing the programs are still beholden to governments and stuff like that. Or okay, what was the impact? How did this work? Was our money well spent? So it's coming up with ways to measure that is a whole challenge in itself. And you can do it with citations, for example, but that has all its own problems, and misses out the fact that you have a tool that's being used in every public health lab, but is not being used for publications, for example. Okay, that's a fun problem. Okay, here's a database and curation related question. Now that we're on that topic, what is the difference between a virulence factor and an AMR gene? Why aren't AMR genes virulence factors? I'm looking at you, Emma.
Oh God. So why aren't AMR genes virulence factors? Well, that's a good question. I think the topic of virulence and the semantics around that are I think Kara mentioned this earlier, very contextual, right? So it depends on the bug, it depends on the environment, it depends on the genetic determinants, and how all those three things come together. So what is a virulence factor versus a factor that affects virulence? I come at this from the fungal world, and people get really polarized as to what what constitutes a virulence factor. So for us, I was working on Cryptococcus neoformans, and one of the three kind of canonical virulence factors is that it grows at 37 degrees. There are lots of things that grow at 37 degrees, and that doesn't mean that they are pathogens, right? So it's really contextual. And so I think ironing out the semantics for that is going to be really important. I know that's one of the I know with we do an ontology called GenEpio, and we've worked with the MacArthur lab and the folks who do all the card curation. And we have started, well, mostly driven by the MacArthur lab, have started to try to put together a sort of proto-virulence ontology to start wrangling these different things. But depending on you're always going to upset somebody. Right, when you're starting to make these designations and classifications. So I can't answer that question for you right now. But hopefully in the coming years, when funders start to recognize the importance of standards and these kinds of semantics for making these kinds of classifications and creating these kinds of knowledge hubs, I hopefully will be able to answer that a little bit better, but I'm going to wimp out and say I can't answer it now, but hopefully in the future.
Inês?
Yeah, I just wanted to jump in to say that you do have a database focused on virulence factors, the VFDB. I can drop the link in the chat. They do offer a definition of what a virulence factor is as something that aids this infection and or disease. So it's a different mechanism to say, like an antimicrobial resistance gene provides resistance to treatments. This one facilitates either the adhesion of a cell to a toxin. So they do provide a definition, and there is a database that contains. How well is it maintained? I am not sure. It's been a while since I've used it, but there are resources there that are focused on the virulence part of it. And one thing is that they do separate by taxa. So for each species, they do have a list of what they consider the virulence factors. For example, my lab works a lot with pneumococcus, and one of the most important virulence factor is actually its capsule, which is not really one gene, is a collection of genes, and they can alter a lot. But yeah, it's very dependent on what organism are you actually talking about. But there are resources out there.
Yeah, definitely. That's a great point. But yeah, you do get into messy territory where, okay, if there's presence of a gene which makes this bacteria grow slightly better and grow faster, more robustly, which means it can grow fast enough to kind of deal with a low level of antibiotics being present in the system. Is that an AMR gene? Is that a virulence factor? Is that just bacterial metabolism? Like it's the classic fuzzy categories that we always end up in life sciences. Yeah, Mark or Andrew, both of you.
Well, when I have a student, a PhD student going into their viva, I always say to them, it's a doctor of philosophy that you're doing here. And you will get asked these questions in your viva. What is a virulence factor? And there are no simple answers to this. It's a very philosophical issue. So I think that, there's a whole question of, what is anything? Is philosophy 101? What do we mean by words? It's a very deep issue, actually. But it's a fun one to think about as well.
So just going back to virulence factors and all that, I know with Tradus, we've done a lot of work where we've taken, one bug, say E. coli, and then done different MICs of different antibiotics. And then it's quite interesting to see which genes become essential and which ones are not as you stress it more and more and more, and it changes. So it's not like you have one set, of genes that it's essential to live in a particular MIC of an antibiotic. It changes depending on the conditions, because bacteria, they're fighting for their life, and they'll do whatever it takes to survive. So there can be different there can be different answers in different scenarios.
Yeah, okay, now that we've done a bit of philosophy, there's a reason ontology is a field of philosophy and something that we use as a tool in this area. So there's some kind of specific kind of questions, and I was lots of people have lots of expertise doing these. So first with I'm sorry about that, I'll close the door a second. We have with these large databases now, and we have large amounts of bacterial genomes, especially, including many E. coli. What do pan-genomes, core genomes, accessory genomes have to do with resistance? How do they differ by different resistance mechanisms? I don't know if anyone wants to jump in and using the use of pangenomic methods in AMR.
I suppose I know a little bit about pangenomes. I don't know much about AMR. But what you do see is you can see very clear patterns of mobile genetic elements, jumping in and out when you look at a pangenome. And quite often those are the bits where you have the resistance genes. So obviously not point mutations, but it's quite a good way to identify not just say the AMR gene, but the structures around it and what other genes are there and what it's coming in on. So you do have to look at sometimes look at the bigger picture, because these AMR genes don't magically appear out of nowhere. I just say we have another special guest joining the panel, Duncan MacCannell from the CDC. So hello, Duncan.
Hey all, sorry to be late.
I have just a question back for you, Andrew, actually, on that point of pangenomes. So it's always been interesting to look at the accumulation of genes in the pangenome as we sequence more and more genomes. Do you know of any research which has looked at the sort of the pan resistome and how that changes as you sequence more and more genomes, and how do we see species jumps occurring as within the pan resistome as over time as we see more and more bugs sort of getting together in hospitals, for example? Are we seeing more things jumping across the species barrier?
I've definitely seen stuff jumping species barrier, and it's only because we've gone and people like the FDA and CDC have gone and done a lot of random surveillance around the world, and sequencing every sample they seem to get and incidental samples that we find it. So our one case I remember was we had XDR typhi, Salmonella typhi, and then looking at the plasmid that it was on, looking back into databases, we found this is in E. coli from cattle in some random place around the world, and there's one little gene difference, but it was more or less identical. And so this seemed to be the kind of ancestor. And it's only by doing all the surveillance that we could actually see that, which is quite interesting. And you wouldn't necessarily go and look for that if you're building pangenome, because you might be looking at like a species or a collection of maybe subspecies, but you wouldn't necessarily be going on a fishing expedition. And it's only using these kind of broad scanning tools that you can find this kind of stuff, which is super cool. I found that using Tsang had a lovely tool. I can't remember what it was called. Big Z
by Fail.
Yeah. And that was quite useful because you just bang in the plasmid and out comes the answer, which is this came from cattle. It was E. coli in cattle, jumped into humans, making crazy resistant Salmonella typhi, which is quite scary.
Okay, another kind of theme that's kind of come up a few times is, and I think Duncan might have some input on this. I'm going to put him on the spot, revenge for being late. How do you, if you're looking at AMR across multiple different environments, sectors, you're One Health, you're looking at in the environment, in agriculture, in clinical settings. How do you choose how do you go about doing that molecular detection and surveillance? How do you choose which tool to use in different environments? Do you try one size fits all and do metagenomics for everything? And what are the kind of trade-offs and considerations between different detection methods and different environments?
I think that's the question, isn't it? I think, unfortunately, it's kind of like how everyone has a favorite tool. Many times the workflows that people put in place are built out of a sense of expediency and around a specific set of applications. Oftentimes, the focus on that is fairly narrow and short term. You're not necessarily considering some of these broader environmental and One Health implications. And oftentimes, with the sheer abundance of choice, I think it becomes a challenge when you actually try and adapt a workflow that has already been established for a certain range of pathogens in a certain context to be able to handle multiple different scenarios. So that was a real non-answer, but I think that is the truth. I think different groups approach it based on the approaches that they have, or the tools that they're familiar with, or the best solution that they come across, and then thoughts of how to actually incorporate that and harmonize that with other forms of testing, with functional susceptibility data, with clinical data streams, other pieces of information. That tends to be afterthought or post hoc, which is challenging. I think that is changing now. I think these kinds of conversations are absolutely critical to talk about how these approaches can be harmonized across multiple sectors, how different groups are approaching it, and how we can overall improve what we're doing.
Yeah, that's great. Yeah,
I'll get something then.
I say it depends is one of the answers. So, okay, you've come up with some way that you're doing surveillance across environments, or even within one environment, just lots of different places. And you've gone through this, and you've done your genomics and got your AMR genes. How do you detect whether an AMR gene has actually undergone horizontal gene transfer or not? And I'll let anyone jump in on this one, because I know pretty much everyone here has done something related to HTT of AMR in some respect. Come on, you want to.
Maybe Henk can come in here.
Yeah, you're muted, Henk.
Yeah, that's the Zoom. Can you repeat the question? Because I was reading the questions of the participants. So that's part of the problem. Yeah.
How would we go about detecting whether an AMR gene has undergone horizontal gene transfer from genomic data?
Ooh, from genomic data. That's kind of, I would say that's kind of difficult. I mean, I'm just thinking about something like multi-drug resistant. So those megaplasmids that you find in Enterobacteriaceae, and if you look hard, you will find them in many genera. It's not very easy to put any directionality on it. Like if we have lots and lots of data for some of those plasmids for Salmonella, because we have 150,000 genomes in the, or even more in the sequence read archive right now, but we have like only tens of genomes sequenced for other genera that are maybe environmentally, not clinically important. So, and I think if you search for Enterobacteriaceae, if you search hard, you will find that they share a lot, and directionality is not really a big one. But one thing you could potentially do is see if there is a case of recent horizontal gene transfer and try to infer, for instance, like phylogenies of plasmids. But again, that's difficult because plasmids are usually like basically a amalgam, like a composite of insertion sequences and your plasmid your functional plasmid backbone, and etc. So that's also difficult. So directionality is a very interesting question, but I think it has to do with database sizes and the overrepresentation of certain species, mainly clinically species. Yeah.
Great. Inês, do you want to jump in on that?
Yeah, I would say like one of the hardest things to know is context. A gene usually is going to be very well assembled, but if it's surrounded by variant or by repeated regions, usually a plasmid-mediated resistant gene is, as Andrew said very well, you're going to need other technologies to actually have the context or where that gene is actually located. And if you know the context, if you know, I know that this gene is surrounded by this repeated regions, this is most likely a plasmid-mediated resistance. It's still very hard to know where the direction of the transmission was. So I say, like, having a close look at what context those samples were collected. I remember, for example, while I was in Groningen, there was an outbreak that was mediated by a plasmid. And it took a lot of work to know where the samples were collected from a patient or from the bed. And sometimes, you know, it's very hard for bioinformaticians to sometimes be aware of the context of what samples are collected and in what conditions and in what time. But alongside having the right technology to get the context, the genomic context, having the right context of how the samples were collected and metadata on the timings, it's really important to try to paint a picture of what is happening. And still, it won't be 100% sure picture, at least not in my experience.
Anthony.
Yeah, I totally agree with what both Inês and Henk have said. Just another thing, I suppose, is in the golden era in the future, when we sequence a lot more, if you're constantly sequencing your hospital environment, then you're able to then sort of track to see where these acquisitions and losses occur within the hospital. Obviously, it's much more difficult out in the community, but at least within the hospital, then you have a greater chance of comparing the chromosomal-based phylogeny with the plasmid-based or mobile genetic element-based phylogeny, and you can perhaps identify those events more easily by looking at those two together.
Great. Okay, I think we might.
I'm sorry.
Okay.
Just adding to that, I would say, like, having, I'm sorry, something is really loud outside. Having the context of the One Health, you know, because transmission doesn't happen just in the clinical setting. Transmission can happen in any sort of setting. So the One Health approach to looking at resistance and transmissibility is incredibly important. And although it at least a few years ago, it was getting sort of good funding, you know, everyone could write a One Health approach problem to figure out the resistance of something. You know, things have changed, higher priorities have arisen, but it's still something that I feel like we should keep pushing forward, trying to have the most complete picture of, you know, it goes from food to people to health.
Great. Okay, we're going to have there's 7 open questions. We're going to have a quick fire round. So just jump in immediately with the answer, and then Mark's going to give a couple of words and just to wrap up as we get towards the end. Okay, which tool would you recommend for Compilobacter, especially using long and short read sequencing data?
AMR Finder.
I guess I'm supposed to jump in and say AMR Finder Plus, but we did do a good job with a whole bunch of Campylobacter. So unless something changes radically, I don't think you'll miss things. But the other tools probably work pretty well too.
And if you're interested in Compilobacter, I know Andrew's podcast, Mike the Microwave podcast, has done a whole episode about Compilobacter genomics recently.
Yeah,
Oh, sorry. How best can we detect AMR genes from wastewater using qPCR? What CT to look out for? And how do we design primers as well?
That's a very difficult task. I mean, it can be totally degraded, you know, and you're going to get bits and fragments and tiny little, you know, is it really AMR? I know some people have done studies on, like, say, the number of vancomycin genes you find in wastewater and the distance from the local hospital, which is quite shocking. And, you know, there is a correlation there, unfortunately. But I think that's a huge, difficult task.
Are there any good resources anyone knows for the design of AMR specific primers? I've done some work evaluating how well current primers work, which is terrifyingly badly. Many of even the UCAS panels are not great. I don't know if anyone's done any work in that area.
So the tool we did with that we have MicroBiggie, which allows you, and it is biased towards the dominant organisms we've been talking about. We do have a way that if you can identify contigs that have those genes, and then you can get sequences with flanking regions around known genes. And we built it for that specific purpose. So that is a way at least you can get a good handle on the flanking regions for developing primers. But then, of course, you have to go make sure that those primers actually work. So, I mean, that's a way to at least And then that was actually by request from people who work on beta lactamases who said we want to do that kind of fishing expedition. So there is some of that, but of course you're limited by, we can only point you to sequences that we already have. But hopefully that will help you get some at least broader primers from, and this is just primer development.
Great. How do we interpret AMR genomic data, which indicates that the isolate would be resistant, but when we do phenotype, we find out it's susceptible? Is that organism sensitive or resistant? Yep, therein lies the great mystery, I think. And there's the interesting research question is, okay, if it has this resistance gene, why isn't it resistant? Does that gene require a compensatory mutation to be effective that our model hasn't captured? That's where I think this real science comes in, the real interesting paper. You get to dig into that. Duncan, do you want to jump in?
I just was going to add, I think the truth, I mean, that's Schrodinger's genotype, right? Resistance or not. But, I think it really comes down to functional results. I mean, clinically, what you're able to actually see in terms of phenotype, in terms of clinically, that's what matters more than the genotype. The genotype, there could be a lot of reasons that could explain what you're seeing, why the markers are present, and yet why it isn't necessarily expressing in the way that you expect. This is one reason, though, why, not to put another plug in for NCBI, although they are fantastic, why I think it is important, the addition, their ability to now host functional susceptibility data alongside SRA records is a wonderful addition, because it actually allows you to tag on that phenotypic information about the isolate in addition to the raw sequence from the genotype.
Yeah. I'll say genomes are capacity, not phenotype. There's always going to be a disconnect between the two. So I don't think, there was a question in the chat, AMR genomics is not going to entirely replace bench work. It's a tool that assists bench work. It can guide bench work to more interesting questions and interesting problems, but they are mutually complementary. One doesn't replace the other. Which tool would be recommended most for TB using short reads?
Okay, I think I have to say that TB Profiler, which I featured in my presentation, I think it works for short reads. So it would be at least my first step. I know that the database is very targeted and comprehensive for TB resistance. So I'll pop the link in the chat. And I would start there at least.
There's also microbe as well, which is similar process, and both are supported by harmonization as of this week. So you can use and compare across tools. Okay. What are the limitations you see to bait capture and targeted capture of AMR for metagenomics?
Okay, maybe I can jump on this one as well. Okay, I would say this is true. If you do just random shotgun sequencing on metagenomics, you'll only know what you'll know. But a database with AMR is a lot more comprehensive that demand how many primers you can create and implement in a multiplex in a simple step. So I would say that you will still going to be very limited on how many AMR genes. So if you're very interested in a particular resistance, I would say it's much cheaper. It's very sensitive and it's wise. But if you want to have a global picture, I would say shotgun sequencing is still, the best. You'll be biased with what you have on your databases, but not as much as you would be if you try to do a PCR panel for it.
Yeah, you're bringing those a priori biases in your design for the baits to your metagenomic experiment, which can be great. It can get, if you have a good idea of what you're going to expect to find, that can be a great way to improve that data and target it a bit more. But yeah, you can also lose things you weren't expecting. Okay, last question. 62 questions. We got through them all. Buying a small sequencer, such as a MinION, for clinical microbiology applications, so detection, AMR detection, outbreak detection, what are the kind of pros and cons?
Well, don't forget about the service cost, right? They can be expensive to run and to get people trained up on them, but they are good for quick analyses. But I would say buy a MinION. Why go with you know, something where you must run a particular batch size to get a cost-effective number of samples through it. If you have a MinION, you can, you know, do a few samples, wash flow cell, do it again, off you go.
Okay, there's some questions that have emerged since I got over ambitious about saying we got to the end of the question, but I think as we're getting close to the end of time, Mark wanted to kind of have a few words to wrap up, so I'm going to pass over to him now. I'll try and answer in the questions.
Great, well, thank you. And thanks for indulging an old man on a Friday. Most people who know me know I'm quite outspoken, but I actually do have an idealistic and sentimental side. And I want to just step back a bit from what you've been doing today. And I'm just going to share some slides with you now. Let me just see if I can get this to work. Here we are. And optimize that and share. So many of you have been dealing with the details of issues today, and a lot of people are going to have questions that they haven't had quite answered. But I always like going back to this quote from Plutarch, one of the ancient Greek scholars, who said, "A mind is not a vessel to be filled, but a fire to be kindled." And I'd hope that what we've done today is actually get people excited about the potential of bioinformatics in AMR research, and that you'll go away and want to know more about this and learn more about this. But the other thing that I came across a couple of years ago was looking afresh at an old saying about the internet, which is that distance is dead. But basically, bioinformatics can be done anywhere. And this struck me when I visited Palestine a few years ago and came across this initiative in Gaza, where people were learning to code in Gaza and actually making good use of their skills in an environment where it's very difficult to get anything much done economically or be productive. And I'm just going to share with you a young lady speaking about her experiences there, which I think can be generalized, because today we've had 58 countries, people from 58 countries participating. We've had participation from every continent on the planet apart from Antarctica in this workshop. And let me just share with you something which I think is maybe seen as particular to Gaza, but I think is actually universal.
صراحة أنا حاولت إني ألاقي وظيفة في أماكن تانية، بس وضع البلد كتير صعب حاليا، وفرص العمل يعني ممكن نقول إنه من 50 حد ممكن 2 أو 3 بيشتغلوا بشكل ثابت، والباقي بيشتغل بشكل مؤقت، وجزء منهم برضو ما بيشتغل بيحاول. وهي هاد من ضمن الأسباب اللي خلتني أرجع للبرمجة من تاني، إنه أنا أوريدي دارسة هندسة حاسوب، وكنت بفكر إني أشتغل بإشي تاني غير مجالي، فلما لقيت إنه سوق العمل مش موفر لي أي وظيفة فرجعت تاني للبرمجة، وبتخيل إن أنا بقدر أشتغل في بيتي وبقدر ألاقي وظيفة من أي مكان. لو كنت أنا بعرف منيح كتير اللي هو السوفت وير سكيلز، وإن أنا عندي هي إنه أنا قوية في مجالي، ممكن أقدم على أي موقع وأحصل على وظيفة بسهولة.
And let me share some knowledge, some wisdom from a friend of mine from the Gambia, who we interviewed recently.
So when I always tell people, when you're looking for the next big thing into what you do with your life, you actually look what would impact you, your surrounding, the society, and the world. And at this moment, I don't think you need to be blind to understand that if you are in the field of microbiology with a bioinformatics knowledge and with being able to do sequence, you have a job for quite a few years. You are in demand. And so if somebody's not convincing you enough, is that there is a niche. There is a niche, and it needs to be filled by people like you. So you should be actually giving your little finger to make sure that you get a program where you'll be able to combine all of this, what I call future as in now, that will give you a niche for you to be able to apply. You would have influences in terms of public health response, influence in terms of how people do with outbreaks we're going to get. And especially in our region, we just, after this one, we'll expect, we're talking about Marburg, we're talking about Ebola coming back. So there is so important to have microbiologists, very trained microbiologists in more modern techniques, that's sequencing, bioinformatics, to be able to deal with this problem. So you could be one of them.
And then I'd like to just leave you with a quotation from my own country, from Joseph Priestley, who was a member of the Lunar Society of Birmingham, which was a group of learned men, included industrialists, people who set up the modern world. And what he pointed out was that they were all united by a common love of science, which they felt sufficient to bring together persons of all distinction, Christians, Jews, Muslims and heathens, monarchists and republicans. And I think that is, to me, that is one of the great pluses of what we've done today in bringing people together to look at this. And it's not just a common love of science, we're addressing a common problem that faces the whole planet. So with that, I'll shut up with my sentimental wittering and leave you to it.
Great, thanks very much, Mark. That was a nice, really nice roundup. So that's us at time. As I say, if you need a certificate and or want to give any feedback for things today, please follow the link that Lisa posted in the chat. We'll also probably send it out possibly send it out by email, as well as notice when we've got all the recordings up and edited and PDFs of the slides, as well as a link to that repository with the training exercise being found in it. So I'd just like to thank again all of the panelists and all of the speakers that took time out of their day to come and help put this together and participate in this. And I hope to see you all in probably and hopefully a future workshop, because it's clear we have a scope for an entire workshop on horizontal gene transfer and on metagenomics, just from questions and points today, possibly one entire one on phenotype from AMR genomics. So plenty of opportunities in the future. Okay. Thank you all very much. Have a great day, everyone. Enjoy your Friday.